Good evening, everyone, and welcome. Thank you so much for joining us tonight for our webinar, Hope After the Standard of Care, Navigating Clinical Trials for ALS. My name is Rumana Barudi, and I'm with ALS United, Illinois, Indiana, and Missouri. We're very pleased to have you with us, and what we hope will be an informative and practical discussion about clinical trials and research opportunities in ALS. For many individuals and families affected by ALS, the world of clinical research can feel both hopeful and overwhelming. Questions often come up about when to consider a clinical trial, how to find appropriate studies, what eligibility means, what participation actually involves, and how to weigh the potential benefits and burdens. Today, we hope to help make that process a little easier to understand. Before we begin, I would like to go over just a few housekeeping items. Everyone will remain muted during the presentation to minimize background noise. We definitely encourage questions, so please feel free to submit them at any time using the Q&A feature. You don't need to wait until the end. We will collect your questions throughout the presentation and address them at the end of the Q&A session. Please keep in mind that today's presentation is intended for educational purposes and should not replace individualized medical advice from your healthcare team. Clinical trials, eligibility, and treatment decisions are very specific to each individual. This webinar is being recorded, and the recording will be made available in a couple of days. And now it is my pleasure to introduce our speaker, Dr. Nathan Carberry. Dr. Carberry is an assistant professor of neurology at the University of Miami within the neuromuscular division. He is a clinician with a background in bioengineering and has both clinical and research interests in neuromuscular diseases and electrodiagnostic medicine. He serves as both principal investigator and sub-investigator for several clinical studies and trials in ALS, as well as in other neuromuscular diseases. Dr. Carberry is also the founder and director of the University of Miami's first genetic treatment intrathecal infusion clinic for ALS. We are very fortunate to have him with us today to help us better understand the clinical trial landscape and how individuals and families can navigate research opportunities. Dr. Carberry, thank you so much for joining us. I'll turn it over to you now.
So, like Jumana was saying, my name is Nathan. I'm one of the neurologists at University of Miami. And the goal of my talk is really to go over the basics of clinical trials. And again, this does not replace the most important advice, which should come from your neurologist in the multidisciplinary clinic. I do have some disclosures. I participate in some clinical trials, largely in ALS, but also some other neuromuscular diseases. I'll mention a couple of these at some point in the presentation because they may be relevant because they're active trials in ALS. Some outline discussions. So I am going to talk about the basics of ALS. That may be a bit of a redundance to some people, but I think important to lay the groundwork. I am going to talk about the standard of care because to me that's really very important to lay the foundation before venturing off and kind of experimenting with clinical trials. So I do want to make sure you're aware of what those are and what justifies them. Then we're going to spend the largest portion of today talking about the clinical trials, expanded access programs. We're going to mention supplements because that always does come up, and I think it's important to be aware that there's evidence for some of those as well. And then finally, the recent advances in ALS and some horizon therapies to keep an eye on. This isn't definitely not meant to be comprehensive, but kind of an introductory course into how to approach clinical trials as a patient with living with ALS. So just an introductory couple slides. So this disease, as you all well know, was first discovered actually in the 1800s. Of course, it's been present for far longer, but Charcot described it for the first time in 1869. It's a disease of two main nerves, the nerve that connects your brain to your spinal cord and the nerve that connects your spinal cord to your muscle. Charcot derived the name ALS, which sounds very complicated, but actually, like many things in medicine, when you break it down, it's not so complicated. So, amyotrophy means that there's muscle loss. Myo means muscle. Atrophy means getting smaller. And lateral sclerosis is referring to the abnormal hardening of the lateral cortical spinal tracts in the spine. So those are just that really was based on a description of the pathology that led to the name. You can actually see it sometimes, not always. It really depends on how much of an upper motor neuron predominance there is to an individual's phenotype. But this is actually an image of someone who has ALS with a really terrible upper motor neuron manifestation of it. So you can actually see this brightness, kind of highlighting the anatomy of the pyramidal neurons extending from the cortex down into the spinal cord. I think importantly, the pathology is something that we're really focused on in terms of finding and applying therapies to stop the progression of ALS. So I think a really important one to be aware of right now is TDP-43. I'm sure you've heard of it. So the neurons in the brain and spinal cord normally have this in the nucleus. In most ALS and not all ALS, and some other neurodegenerative diseases, the TDP-43, for whatever reason, wiped out and can be found in the cytoplasm. So this is something of significant interest in development of therapies in ALS. And there's really a chicken or the egg kind of theory for this. Is it causative of ALS or is it just kind of a biomarker telling us that, hey, ALS is here? There's many drug targets, including TDP-43, some of the structures that surround the normal functioning of this protein, in addition to many other things, which we're working on targeting and also using as biomarkers to measure a treatment response in ALS. There's so many theories on why ALS happens, which is why there's so many ongoing clinical trials throughout the country and also in the world. We definitely don't have the answer yet, and that's why things seem so complicated because, like anything else, when we don't really know what's going on, it can seem very opaque. But what I think we all agree on for the most part is that there's probably some incipient problem that leads to a downstream effect of damage, and maybe it manifests with excitotoxicity, neuroinflammation, mitochondrial dysfunction, or what have you. And we want to get as close to the starting point of the disease process as possible so that we can benefit from all the downstream effects as well. But all of these things you see here are targets in drug therapies and have been associated with certain genetic mutations, things like that. So what are the established therapies? Well, the first main one that came out, and I'm sure many of you have heard of it. If not, you should definitely ask your neurologist because this one has the most evidence currently, is riluzole. So this was based off of a trial that, like a phase 2 study. And the phases I'm going to get into, what the numbers mean and which numbers you should be most interested in as someone living with ALS. But this came out in 1994 and it was very exciting. It led to approval a year later, and it's based on the pathophysiology of glutamate causing excitotoxicity in the brain, in neurons in the brain and the spinal cord. And this is the target of riluzole, and it reduces excessive transmission of glutamate. This particular trial, I'm not going to get too much into the nitty-gritty, but I think important so you understand what we're looking at for clinical trials so that you know what you're getting yourself into if you're thinking about participating. What are the main endpoints that, and this is something you'll see in most trials that output at phase 2 or 3 is a Kaplan-Meier curve where we look at survival. And what we're trying to see in these curves is a separation between the treatment arm and a placebo arm. Placebo is that dirty word that no one wants to hear in any neurodegenerative disease where you're not getting the therapy for a certain amount of time. Anyway, in this group, there was clearly a separation between the treatment group and the placebo group for survival, which is exactly what the scientists back then wanted to see. So it showed survival benefit in bulbar onset and the general population, and less so for limb onset. That doesn't mean we don't encourage everyone to take this if they can. And it reduced mortality at 12 months. And then even at 21 months, it continued to reduce mortality. And these findings have been corroborated over and over again, including a phase 3 study that was performed in 1996. So I'm going to give this one the badge of survival because I do think it helps with survival, and I think most neurologists would agree with that. Radicava is the second medication I'm going to mention before we get into the clinical trials, and because I do want you to be aware of this. This is a new medication that was approved in 2017, so I should say relatively new. It was actually started as a stroke treatment for free radical scavenging. It was a Japanese drug. So this medication, it's a pyrazolone, which is known as a potent free radical scavenger. And so the thought, if you look at that initial graphic, hey, there's a lot of neuroinflammation in ALS, or at least there's thought to be. Wouldn't this make a good drug to treat ALS? And that was the justification. So there was a phase 2 study that was done in, I think, 2014. It was a double-blind study, and it did show a reduced concentration of one of the toxic byproducts in that pathway, which was encouraging, meaning, hey, this medication is reducing inflammation in some way. It's doing what we want it to. Unfortunately, that primary endpoint of this score, which you're going to see over and over again, not just in clinical trials, but also in your clinic visits, where often the neurologist will ask or a nurse might go through a questionnaire, and this is a functional rating system, and it's very often the primary endpoint, meaning the main thing we look at when a clinical trial is performed to see if it worked. And this is really measuring a functional decline. And we want to see it stabilize, or, I mean, miraculously, could we see it even improve? Anyway, so for this medication, which is available now, it did not reach its primary endpoint. And so we're wondering, you in the audience are probably wondering, why is it available? And the reason why is because in this fairly large group of patients, fairly heterogeneous group, when looking at a specific subgroup within that larger cohort of early stage ALS, meaning people that just started having ALS, it actually did show some benefit. And it was mostly with regards to function. So I'm going to give it a half badge of function for now. And there was only one secondary point which showed any significance. And then there was another trial, like a follow-up study, which did show significance in that point when looking at just that group of people that are early on in disease onset. That kind of solidifies the bench that, hey, it does seem like it helps with function. And that's why it's recommended especially for those patients early in their disease course. And so that might be why a neurologist may or may not have offered you this drug, depending on when your disease started. And then just some additional details. There were a couple of secondary endpoints that were positive, and there has been a more recent study, a phase 3, which is a larger study, which was a little bit mixed in the results. But currently, riluzole and radicava are recommended in the clinic. So that's your ground floor. So whenever I'm introducing the topic of clinical trials, I always make sure that the house is built. You want to have some structural foundation of evidence-based therapy before you start thinking about experimenting with new medications. Because of course, we're not satisfied with where we are in the treatment of ALS. Otherwise, we wouldn't have all these clinical trials. You wouldn't be sitting in the audience. But I do want to make sure that's clear. All right. So now the whole reason why you're here. So clinical studies and clinical trials. So generally, for just the nomenclature, study, what that is generally referring to is an observational investigation where we collect data. I will say that this is not usually the type of investigation that patients are seeking. It's usually done in conjunction with a clinical trial where we do interviews, surveys, examinations, nerve and muscle tests, the dreaded EMG, biological specimens. That means blood or CSF, cerebrospinal fluid. These are really because they help us understand the target disease and help us find new biomarkers and new targets. Clinical trials are what you're here for. They're to investigate new experimental treatments. because you get started with these trials, maybe you know, uh, a goal to help the general population, but everyone that is participating in clinical trials hopes that it might benefit you as the person putting in all the work and involving in meeting the scientists, these doctors who you'll see so frequently. You want to benefit from the clinical trial. So this is really what everyone's seeking, and I will just encourage that you can enroll in one or both. So you can do as many studies as you want. The clinical trial usually one at a time, and I encourage you to do both. All right, so clinical trials. When did they start? So really, the first known modern clinical trial was in the 1700s. It was a treatise of the scurvy, where some sailors got oranges and lemons, and they didn't get scurvy. At the time, they thought it was because of a digestive issue, although it was vitamin C, turns out. But anyway, that was the foundation for a clinical trial. Ethical codes really weren't a thing until the mid 1900s and then the 1960s. It's all based on this clinical equipoise, meaning, hey, we have a problem. We don't have the answer for it. Could a new medication help? There's 5 phases to be aware of for clinical trials. And this is a graphic that I made, which is meant to be a metaphor. So way up in the top left, we have the preclinical trial. And there's no humans involved. This is the creek. This is just the water trickling by the pebbles that maybe with many contributions of many creeks makes a small river, and that's phase one. And there's far fewer rivers than there are creeks, and there's far fewer rivers than large rivers than the Mississippi, and that's phase two. So they get bigger and bigger. Phase three is a lake, and phase four is the ocean, dumping out into the ocean. So I want you to be aware of when you're looking at clinical trials, where are you on that trajectory? Are you way up in the mountains with with the creek? Probably not, because you're not an animal, you're a human. But maybe you're in the tiny river where you're just barely afloat. So I want you to just be. I'm going to go through each of these separately, but this is the metaphor that I'm going to carry through the whole presentation. So preclinical, like I said, is animals, and that's usually rodents. For ALS, there's a few different rodents that are used in all sorts of labs all over the world. Many of them have certain genetic mutations which may or may not represent your ALS. I always say everyone's ALS is different. One person's ALS is not the same as another person's ALS. So these preclinical investigations, you take them with a grain of salt. They have not been, there's no human has taken those medications yet, exciting as they may seem. Phase one is the first in human. So that's that tiny river here. And I'm putting a little beaver dam to kind of put a strong separation between phase one and phase two, because phase two and onwards are really the ones that have the benefit to help you. Phase ones are really important, but phase two, three, and four are the ones that are most likely to show a benefit for you. And through all of this, I'm going to remind you that if you're thinking about doing clinical trials, you have to have your foundation. You should have a stable, known evidence-based therapy in place, and that means Riluzole, Radicava, and then even a couple new therapies, which I'm going to talk about at the end. All right, so the preclinical investigation is what I'm going to start with, and this is probably what you hear about the most, on the ALS today. Maybe a very exciting new cure for ALS showing unbelievable promise, and then when you actually read the article, it's just in rats or in you have to be aware of not a mouse. These are very important because they evaluate drug production and purity. They explore safety and doses equivalent to approximated human exposures. They test pharmacodynamics, meaning the mechanism of action and the relationship between drug levels and clinical response. They measure pharmacokinetics, meaning absorption, distribution, metabolism, excretion, drug-drug interactions. They'll test all sorts of stuff to see if it's safe in a rodent. It's the most common and least expensive clinical trial step. So again, this is where you're going to see a lot of very exciting data come out. But remember, you have to remind yourself, where are you on that journey? And these are all the way up in the mountain. They have to come all the way down into the river, the lake, and the ocean to be really really exciting. So I do want you to be to beware really, kind of this puddle news of sensationalism about finding the cure. I think so critical to be hopeful because I am extremely hopeful that we're going to find something that will cure ALS, and I know that my colleagues all feel the same. There's definitely a positive energy, but I really want you to be aware of that. To just and always feel free to ask your neurologist a question of, "Hey, I heard about this new treatment, and what do you think?" And most neurologists would be very happy to learn about a new treatment because there's so many coming out all the time. All right, phase one. So the question with phase one is it safe in humans? That's really the main question. It's also called a dose escalation or a human pharmacology study. So notice I didn't ask the question, is it beneficial? That's not the question. The question is it safe? There's no potential for benefit to the participants, unfortunately. It's usually performed open label in a small number of healthy and or diseased volunteers. So a control will get it, and a person living with ALS might get it. And they're both getting the same medication, maybe in different, varieties, different doses, different combinations, all sorts of things they might look at. And it's a small number of people. And they explore, again, the safety, the maximum tolerated dose. So they'll escalate until we start getting adverse effects. The same thing, the pharmacodynamics and the pharmacokinetics, drug-drug interactions. And the ones that are successful, that seem safe, they pass the dam. They are able to go through, jump over the dam, pass under the dam, whatever metaphor you want to use, into phase two. So phase two is where we start to get excited, where we say, hey, this drug has some early signs in the preclinical studies, in preclinical trials that it seems safe, that there's some signals that it might work, and if the science seems justifiable, then we're on board, and we want to support these things and get our patients the opportunity to participate if they'd like to. So these are also called therapeutic exploratory trials, and the main question is, can it work? I'm not saying does it work. The question here is, can it work? Is there a strong enough signal here that tells us we should do even a larger trial, which is the phase three. The phase two is, does it seem like it could work? It's not powered, meaning there's not enough patients that participate in these to establish the true efficacy of a drug. It's possible to get a drug approved in the phase two, but it's not the expectation. So it's conducted in patients who have the disease of interest. Some subjects are randomized to a control arm, and blinding may occur to limit assessment bias of subjective outcomes. That's a word that we use a lot. What the heck does blinding mean? So this is the way that the trial is structured. So everyone has ALS. Some people get the placebo, and some people get the drug. With blinding, so there's single, double, and triple. What we're referring to is who knows who's getting what. Who knows what patients are getting the treatment and which patients are getting the placebo. So in single blinding, which is not that common, the investigators will know, but the patients will not. Double blinding is the most common, where both the participant and the investigator do not know. And it's let's say it's an infusion with a bag. It may have coverings or dilutants or something like that to make it look exactly the same. So really, the sponsors who make these medications do a very good job to protect the sanctity of that, because it really does affect the purity of the results. And it's almost like a religion. We really try to respect the blinding of these trials. So double blind is the most common, and what that's referring to again is that the investigator and the patient do not know what they're getting. Triple blinding is when you add in the data analyst also not knowing, and that's not as common. All right. So what are these for? So we explore again safety. We're always looking at safety, and that becomes more and more apparent as we expose more people to a new drug. Because you might imagine if we just start off with like 5 or 10 patients, probably we're not going to see all the adverse effects that you can when we start seeing tens of thousands of people getting the medication. Always the human pharmacodynamics and kinetics. And then really the question here is what will justify a much more expensive phase 3 trial? So optimal doses, dose frequency, administration routes, and endpoints. So biomarkers, signals that, hey, this drug is reaching its target, it's doing something. And I'll talk about some recent successes in ALS where a phase 2 trial really showed very strong biomarkers and that led to early approval. So this can sometimes lead to something called accelerated approval by the FDA, which again is not common, but that's in situations like in ALS where there's people are desperate for a new treatment, and there's you don't want to wait years and years for a new trial where something like this might happen. All right, phase 3. So this is the ones that if you can participate in a phase 3, that would be the best one to participate in probably. Phase 4 I'm going to mention at the end, but that's far less common. Phase 3 means that hey, there's good data to show that this is probably helpful or could be helpful to the extent that the person who made it is willing to fork over hundreds of millions of dollars to basically gamble and see if it might actually show all the signs of being helpful for slowing down the disease in ALS. So if you can participate in a phase 3, this is the one to go for. So this is otherwise called a therapeutic confirmatory, comparative efficacy, or a pivotal trial. So it's much larger. Usually 300 to 3,000 people will participate. And you are really just demonstrating or confirming the efficacy, and you're always looking at adverse reactions. You never want to cause harm. So even if you have the best medication in the world, if it causes a terrible side effect, then it's never going to make it to market. So the more rare side effects are not necessarily going to come up in a phase 3. And that's because you're still only dealing with a few thousand people. That's like a high school. So, when you expose to, statewide or multi-state, then you're going to start seeing all the adverse effects. So, like the other ones, you're comparing a treatment against a placebo or a standard therapy. And there's 3 people in this diagram because it's getting larger and larger. And of course, this can lead to FDA approval of a new medication if it's successful. So, I'm just going to take a moment to talk about a medication which was withdrawn from the market after a phase 3 study or phase 3 trial, which I think just reinforces the role of a phase 3. Like, why do you do it if the phase 2 looks so good? So Relyvrio, maybe many of you don't know about this medication, but it was approved a few years ago and then taken off, a year or two later. And it's a combination of sodium phenylbutyrate, and I'm not even going to try to pronounce the second part. We call it TUDCA. Its therapeutic effect was thought to be related to neuronal toxicity, common theme with the other medications. And the Centaur phase 2 trial performed in 2020 had a primary endpoint, just like the other ones, of ALS-FRS-R, which was significantly improved in the treatment group. But remember, the phase 2 study is can it work, not does it work. So it showed signs that it could work, but it did not prove that it was working because this was not a phase 3. It's a smaller study, smaller trial. And all the secondary endpoints failed. It, however, was approved for treatment in ALS because it's a very I will say that the ALS community is unbelievable. It's very, very powerful. You'd be shocked at how much pull you have in governments and raising money and making big changes in FDA regulation, things like that. So in large part because of the strength of the patients in the community, it was approved because people are desperate. Unfortunately, the medication had a phase 3 study which did not meet any of its endpoints, and it was actually withdrawn by the sponsor. So just to kind of reinforce that, it's important to go through this process. This was not a very toxic medication. It had terrible taste, but that's about it. Maybe it caused diarrhea. But God forbid we put something on after a phase two that's damaging, where a rare side effect ends up being horrific, and then the phase three shows that it wasn't helpful at all. Then you're actually causing harm. All right, phase four. So this is now the end of the this river and lake metaphor that I've created. So this is otherwise called a therapeutic use or a post-marketing study. It's performed on FDA-approved drugs in large cohorts to really find less common adverse reactions, and also to evaluate the cost and/or drug efficacy and populations or diseases or doses that were not tested in the phase three to trials. These are not performed very frequently. Actually, less than half of these trials that are recommended by the FDA or required are actually completed. And you might understand why, right? The drug's already in the market. People seem to like it. Then what's the point? But just to remind you that we may find things later on as drugs become more widely available that might be identified either after the drug is approved or in a phase four trial. 20% of drugs acquire new black box warnings after the approval, after the marketing starts. That's a lot, 1 in 5. And 4% of approved drugs are ultimately withdrawn for safety reasons. So, another really important reason to be very much plugged in with your multidisciplinary clinic, especially if you're thinking about embarking on one of these clinical trials, because it is a risk. You're taking a little bit of a risk. All right. Expanded access program. So I think it's definitely not the focus of today's talk, but I do want to mention that this is an opportunity for those that can't participate in clinical trials, whether because they're person's too progressed in the disease, or there's certain inclusion or exclusion criteria that you just didn't meet, didn't work out for you. These are ways to get access to phase two and phase three medications that are currently being investigated or awaiting the results to see if they might be beneficial. There are not that many of these, but I'd say they're very popular because it's a way to, it's kind of a low barrier to entry for medications that may be helpful. So these are available for really medications within that phase two or three category. And a patient living with ALS may be able to get those. There may be some associated costs. I'm aware of at least one medication, that is pretty popular right now that actually has a cost to entry. The inclusion-exclusion criteria tend to be less restrictive. All right, so the take-homes. So first of all, I always bring up for my patients in my multidisciplinary clinic, what is a clinical trial? What is a study? But I always make the point that, hey, it's not for everyone. Some people, it may be too much. It may be, this may not be how you want to spend your time. Sometimes it's better to stand in the sidelines than to play in the field. It's like watching a soccer game versus, playing in the soccer game. So if you're rooting for your team and your team wins, you can still benefit from that, rather than, kicking the ball and saving a shot. You'll most likely be required to remain on a stable dose of the standard of care. So just be aware of that. So let's say you just got diagnosed with ALS. I would take the time to build your house, meaning get stable on the ribosol, get stable on the Radicava. And there's one more medication which I'm going to actually bring up, which is really becoming a standard of care, which is the B12 megadose. Not to spoil the end, but I'll talk about that in a moment. But those medications, you have to be on a stable dose usually. If you experiment with stem cells, that's usually a no-no for a clinical trial exclusion criteria. And that's just because we don't really know how those might affect the results of these trials. Um, Really important to be practical. Where is the site located? I think that's maybe the most important. Like if you live right next door to, I work at the University of Miami. My first question is always, where do you live? If you live in, the northern part of Florida and you're traveling all the way to see me in Miami, and I can tell that you're someone that's progressing fairly rapidly, I think that's something to be really, really aware of because it may take a toll on you. And you want to look at the protocol. How often will you need to visit? Sometimes those trials require visits, every week for maybe a couple months. Usually it's spaced out after that. But that may be something that you don't want to do. Like, let's say you just hate going to the doctor. You're just always someone who hated going to the doctor. That's really something to be aware of. Or you hate blood draws. Do you need to do a lumbar puncture? Usually this is optional. That's just to give you that secret. Um, it's good to do these if you can, but just be aware that if lumbar puncture is on the protocol, usually there's a get out of jail free way to avoid doing that. But I do multiple lumbar punctures every day as part of my job in research, they're very safe, and I do think that they're critically important. But just there's things like that to watch out for. I think really important is this how you want to spend your time? Do you want to be going to see your neurologist? Do you like your neurologist? That's important. Um, and I think very important for the neurologist to be likable. Um, what is the potential of receiving placebo and for how long? That's a really important question. There's some really important resources that you really have to advocate for yourself. And you can use these resources. The clinicaltrials.gov has all of the ongoing clinical trials in the United States. I would discourage you from going too far outside of your state. Maybe if you live in a tri-state area or something, you can go a little bit of a distance, but I wouldn't be traveling across the country for a clinical trial, generally, unless you're planning on moving there. ALS Signal is another one. This is what clinicaltrials.gov looks like. So if you just load it up, you put in your disease, ALS, and then you just press search, and you can find all the ones that are ongoing. And you can actually read the protocols. Usually the protocol is actually listed there, and you can start reading about them. I would have a very low threshold to ask your neurologist for advice about this. Don't be shy. That's their job. And if they don't know something, the best neurologists will not be ashamed of that. They'll actually work with you, shoulder to shoulder to find something that might be good for you. Because we don't know everything. No neurologist knows everything. This is ALS Signal, which I really like. This is a fantastic dashboard. I use it all the time for my patients, which is not just the United States. There's clinical trials that you can look at outside the country. You can, filter based on, randomization, whether there's an open label extension, all these sorts of things. And you can, find the name of the medication and maybe you want to ask your neurologist about the mechanism. Do they think that science makes sense? Things like that. Route of administration is an important one. So intrathecal. So that means in the spine. So that in my introduction, it was mentioned that I started a genetic therapy clinic. That's to use this medication, that Qalsody. But intrathecal therapies are, I would say, becoming more popular in kind of the genetic bend of clinical trials in ALS. All right. In my last few minutes, I'm going to talk about supplements, some horizon therapies, and some recent successes. So supplements are very popular in the treatment of ALS. These are things that your neighbor comes over with, the news that if you expose yourself to, a toxic level of zinc, then your ALS will go away. Please utilize your ALS neurologist and a resource I'm going to give you at the end here before you start taking these supplements. In general, there's limited evidence to support the vast majority. Some of these can be very toxic, especially if mixed together. I will say that there's a risk of a kind of a popular science exposing patients to a toxic soup of supplements. I really don't want you to make yourself a petri dish because you're not a petri dish, you're a person. and you really should ask your physician for advice. ALS Untangled is the best resource for this. So this is a kind of a conglomerate of contributions from a bunch of neurologists. University of Miami participates, but it's driven by a guy named Richard Bedlack out of Duke, who's fantastic. He's a really phenomenal ALS neurologist, and he really focuses on supplements and the evidence for supplements. And this comes up so often, so I use this website probably every day. Um, and you can just go through and it breaks things down by mechanism, what phase are we in, cases, trials, what are the risks, and that sort of thing. So before you start experimenting with all sorts of supplements, I would definitely use this as the first step, the ALS Untangled, and then also ask your neurologist. All right. So some recent advances, just to give you some hope, because there has been some really fantastic recent advances in the treatment of ALS. So you may have heard of this medication tofersen or Qalsody for SOD1 ALS. So it's an antisense oligonucleotide. What does that mean? So it's basically a piece of DNA, of manufactured DNA, sort of that prevents mRNA or messenger RNA from being translated and producing a toxic protein. So it reduces the translation of this protein, and it kind of slows down ALS in this particular subtype of ALS, which is the SOD1 genetic form of ALS. So the Valor phase 3 study resulted in 2022. Its primary endpoint was a decline of functional status. Again, that's coming up again, ALS-FRSR. So you can see that it didn't quite reach its endpoint at 28 weeks. These lines are overlapping. We don't like overlapping in clinical trials. We want to see these separate. So 28 weeks that failed, but in the open label extension, you can clearly see a separation. And in the patients who were exposed to therapy early had the best effect, but the ones that were exposed to it later also stabilized. So this was very exciting. This also had some really exciting secondary endpoints, meaning things that we looked at besides that functional decline. And this is something that you're hearing about in the news and also in your multidisciplinary clinic, which is neurofilament light chain. So that's a very hot biomarker, which basically represents nerve breakdown products in your blood. And this really showed a strong response in the treatment group, and in the open label extension, these lines combine together, and that's exactly what you want to see. And the other secondary endpoint that was positive was that protein, which was toxic, and we want to get rid of it, and it showed a significant reduction in this curve here. So that led to its approval in 2023, and it's been amazing. There's been a lot of clinical success. I have a large clinic in Miami that I started, and it's been amazing. Actually, some people have had improvement in some of their function, which has never been seen before, really in any neurodegenerative disease. So it's very exciting. It feels almost like the moon landing. Um, and it's just been fantastic. These are some other endpoints. Slow vital capacity is your breathing, your ability to breathe. So that also showed a stabilization. And look at that curve here, even a little bit of improvement in the weeks 40 to 52. And there's been some publications from Tim Miller, Stephen Smith, looking at the real world application of this and showing actually functional improvement in some patients. So it's really amazing. So this is really some energy to say, hey, clinical trials are worth it. There could be some major successes. And the Atlas trial, which I participate in quite a lot, is to see if we can stop ALS from happening in the first place. And that again is focused on SOD1. But we're hoping that the things that we learn from this trial, which I hope will be successful, we can apply to non-genetic forms of ALS as well. That neurofilament light chain is again coming up, and I won't go into the nitty-gritty of exactly how this trial is run, but just know it's the first of its kind, and we're very excited about it. So some horizon therapies, my last couple minutes, to just leave some time for questions. So I would say that these genetic therapies are really quite popular because of the success of tofersen. Really, that's energizing a lot of neurodegenerative disease research and other things like dementia, because it's really like a major breakthrough. So can we apply these sorts of mechanisms more broadly? Are there other genetic forms of ALS which might benefit from the same sort of tool? So Silence ALS from Dr. Neil Schneider at Columbia is looking at that. It's looking at really rare genetic forms of ALS where really maybe one or two families might be affected and building custom genetic therapies for those folks. We have some local successes in Florida from the Mayo Clinic with Dr. Oscarson with CHCHD10, which is a very rare form of ALS, showing similar results to tofersen with SOD1. There's other genes under investigation, and actually, there's even non-genetic forms of ALS, which we're hoping to use the same sort of technology as a sort of instead of turning a toxic gene off, can we turn a savior gene on or amplify it? Um, all right. So the last thing I'll mention is intramuscular B12. So this one is actually almost so popular that it's become standard of care, although it's not approved by the FDA and you have to pay for it out of pocket. But it's standard in my clinic, and I'm sure you've heard of it. But it's based on the success of this trial from Japan called JETLS, which is a twice weekly intramuscular injection of B12. Same primary endpoint. It was significantly improved in the treatment group, and it's well tolerated and safe. People generally feel more energetic when they take it, and it seems like a good additional medication to use. So I recommend it to most of my patients. I'm not giving you advice. You have to talk to your neurologist, but this is a new medication, which I expect will be approved by the FDA at some point. Prime C is something that I think is very popular. We're still waiting to hear the kind of the final word on how effective this medication is. It's a combination therapy of an antibiotic and an anti-inflammatory. It's a very small study, which led to a lot of, I would say, popular science, publications online, people being very excited about it. But this is one of those ones where we have to remind ourselves that this is a phase two, not a phase three, where we don't know, maybe there are signs of can it work, but not does it work, which is phase three. So this medication is not currently recommended, given its limited data. And that's all I wanted to talk about for now, but I'll have time for questions if you have any. And thank you so much for listening.
Thank you so much, Dr. Carberry. That was an incredibly informative presentation, and I think you've given us a lot to think about. So thank you so much. So we're now going to go over some of the questions. We have a bunch that came into the Q&A, and I had some that came through a different chain. So we're going to start. Andrea asked many questions. Some of them you have already responded live, but I will go over them very quickly. Her first question that was not necessarily answered was, what do classify as early stage ALS?
It's a very, very good question. So whenever you're filtering, whether it's clinicaltrials.gov or ALS Signal, one of the inclusion criteria is going to be, how long have you been symptomatic with ALS? Usually it's, 36 to 48 months, maybe a little bit less, depending on how many patients they want to squeeze or they want to have a larger subset of patients that might qualify. But it's usually, within a couple of years of disease onset. And we are careful in the way that we define disease onset by onset of weakness. So very often when you're in the clinic and you're, kind of racking your brain when the neurologist asks you, when did your disease start? You think back, well, I had a cramp, 4 or 5 years ago, or I had a twitch in my cheek, 3.5 years ago. Those don't usually count for clinical trials. So it's usually onset of weakness. And you can look and see there's a variety of what we consider early stage. But in terms of time, it's usually within a couple years that you might qualify for a clinical trial. And then in terms of function, that questionnaire, the ALS-FRS-R, there are usually requirements of a certain number that you have to be in order to qualify. There's even sometimes where you have to look at the slope, meaning what is your rate of progression of the ALS-FRS-R over a certain amount of time, which might require a couple data points for your neurologist to collect. So, these are very good questions, and there's even additional things that I'm not mentioning. I could go on and on, but hopefully that answers a little bit.
Thank you. So, Andrea's next two questions are kind of connected. They're around the standard of care and research. One of them is how long should you be on Riluzole and Radicava before entering a clinical trial? And then being stabilized on those medication, what does that mean?
Yeah, great question. So that again depends on the clinical trial, but usually like a month, 4 weeks. And a stable dose means so Riluzole, it's dosed morning and evening. Let's say that you started the medication, you got an upset stomach, and you could only take it once per day. So that's fine, but you have to be on that once per day dose for a full month before you can enroll in the clinical trial. The reason why is because if you think about, what are we trying to do with these clinical trials, and why are these sponsors spending so much money on running them? It's to prove that something might work. So what you don't want to do is muddy the picture with all sorts of other medications which might be entering at the same time as the drug under investigation. If that makes any sense.
Okay. And her next question is, what are the risks of lumbar puncture?
Usually we say that this is so safe that we let medical students do them. Um, so with any procedure, and your neurologist has to walk you through this, there's three main risks, um, and kind of tailoring this specifically to lumbar puncture. So the first risk with any procedure is bleeding because you're entering into past the skin, and there's potentially some blood vessels which might be involved. There's risk of infection, right? Because you're entering the skin. The skin is your main defense against infection. I will say I've never heard of anyone getting an infection from lumbar puncture, but it's a theoretical risk. And then for lumbar punctures in particular, that third risk is headache. So when you enter into the spinal space, most likely you're going to be taking out some fluid. And the reason why we do that is one, it's the closest that we get to the spinal cord and to the nerves without taking a piece of those, which we would never do, right? Those are not something you can grow back. But two, for example, in my genetic therapy clinic, this allows us to make space for the therapy that we're going to fill back in. So it's almost like getting an oil change. You take out 15 milliliters of fluid, you put back in 15. So those are the three main risks, and there's ways to mitigate the risk for all of those. And I have patients that get these done every month, every 2 weeks for a certain period of time, and then every month. And I've been doing it for years. So and they're doing just fine.
Can Qalsody be once eventually used for patients without SOD1?
That's such a good question. So at Washington University in St. Louis, they're actually running that trial. I've done it myself a couple of times. Um, the justification for doing that is because some patients who don't have SOD1 in autopsy have been found to have the same toxic forms of SOD1 that SOD1 patients have. So the question is, could SOD1 be involved in all ALS or in a large chunk of ALS? So that question that you're asking is something that we're asking in the scientific community. It's a question I've asked a couple times with individual patients. but right now it's only approved for patients who have SOD1. But I do think that this same sort of model for treatment will benefit other patients that don't have SOD1 in the future.
Okay, kind of related to this, I have a question. Is given your work with genetic treatments, how is genetic testing changing clinical trial landscape in ALS? And should everyone living with ALS consider genetic testing, or even when there is no known history?
So historically, before we had a new therapy that was effective for ALS, and tofersen I will label as the most effective therapy that we have in ALS, even better than ribosol. Um, And so once that medication became available, it became kind of a no-brainer for anyone to who has ALS to get checked for to see if they have a genetic mutation. We actually don't use the terminology familial ALS anymore for this exact reason, because we don't want to discourage people that don't have family history from getting the genetic testing. I have so many patients that have been shocked by their genetic results because they don't have any family history. Maybe they came from Cuba and they don't know why their parents passed away. Or, this kind of a specific example for me being in Miami, but absolutely, it's completely changed the what happens in the multidisciplinary clinic in ALS. And in terms of the clinical trials in general, I think that we're obviously very interested in why did this medication work so well? What can we learn from it to improve other medications? And then also, how do we deal with patients who have SOD1 and they are getting to a first, and can they enroll in other clinical trials? And these are big questions that we're asking, and it's definitely an exciting time.
Awesome. One last question before we close. When you look at the ALS research landscape today, what developments or approaches gives you the greatest reason for hope?
So I think I came at a really exciting time. I feel very lucky because if you look at the rate of discovery in ALS over time, I put a little bit of that in the presentation, but 1994 was ribosol, then there was nothing until Radicava in the mid 2010s. And then there's another medication, Relivrio, that came right after that, and then tofersen came right after that. It's exponential. So the rate of discovery, the amount of investment that's going into ALS research, and then kind of fired up by this major success in a genetic form of ALS. And so we're really proving for the first time that we can take on neurodegeneration. And we're very excited about it, and we're definitely optimistic about the rest of ALS.
Wonderful. Thank you so much for sharing your expertise with us and for the important work you're doing in ALS care and research. I might have another question. I'll follow up via email to share it with the recording afterwards because
Sounds
we're
good.
running out of time. So thank you, and thank you everyone who joined us tonight. And to those of you who submitted questions, we know that navigating ALS trials can sometimes be complicated, but we hope today's conversation has helped provide a clearer understanding of the process and the questions to consider along the way. At the ALS United Illinois, Indiana, and Missouri, we remain committed to helping individuals and families affected by ALS stay informed about research opportunities and connect with resources that can help them make decisions that are right for them. Once again, a reminder, we will share this recording from today's webinar in a couple of days. And thank you for your patience with that. Thank you again, Dr. Carberry, and thank you to everyone for spending part of your evening with us. Have a wonderful evening, and we hope to see you in the future.